So what is DNA replication? DNA replication is the process by which DNA makes a copy of itself during cell division. The first step is to unzip the double helix structure of the DNA molecule. This is carried out by an enzyme called helicase, which breaks the hydrogen bond holding the complementary bases of DNA together. Separation of two single strands of DNA creates a Y shape called a replication fork. The two separate strands will act as template for making a new strand of DNA. One of the strands is oriented in the 3' to 5' direction towards the replication fork. This is the leading strand. Short piece of RNA called a primer produced by an enzyme called primase comes along and binds to the end of the leading strand. The primer acts as the starting point for DNA synthesis. DNA polymerase III binds to the leading strand and then walk along it, adding the new complementary nucleotide bases to the strand of DNA in the 5' to 3' direction. This sort of replication is called continuous. Then DNA polymerase I will bind to it. It will strip away the primers. The gap where the primer were are then filled by yet more complementary nucleotides. The strand is oriented in the 5' to 3' direction away from the replication or fork. This is the lagging strand. As a result of their different orientation, the two strands are replicated differently. Numerous RNA primers are made by the primase enzyme and bind at various points along the lagging strand. Chunks of DNA called Okazaki fragments are then added to the lagging strand also in 5' to 3' direction. This type of replication is called discontinuous as the Okazaki fragment will need to be joined up later. Finally, an enzyme called DNA ligase fills up the sequence of DNA into two continuous double strands. The result of DNA replication is two DNA molecules consisting of one new and one old chain of nucleotides. This is why DNA replication is described as a semi-conservative. Half of the chain is part of the original DNA molecule, half is a brand new. Following replication of the new DNA automatically wind into double helix. Thanks for watching. I hope you enjoyed the video.